Hello, welcome to my YouTube channel. Today we are discussing about the cell cycle and cell division. In this chapter, we're going to discuss first we have gone through snapshots, some important points of the chapters. After that, we look into MCQs practice. So, we go like this. In the universe, every living being has some cells and it produces some another cells by cell division. If you take these cells, which may be like eukaryote and prokaryote, first prokaryote and then you take eukaryote. Prokaryotes, it is like aiims. If you take eukaryotes, there is two types. One is mitosis, another meiosis. After this, if you see the cell, this part is 90% of the cell cycle is constituted of interphase. Interphase is a long phase where cell prepare for its cell division. Only 5% represent M. This M is divides into two types. One is mitosis type of cell division, another is meiosis type of cell division. After this, in my interphase there are three phases where cell prepare for its cell division. In interphase, cell DNA replication is also happen, and protein synthesis and cell actively grows in size. All this happens in interphase. If you look into interphase 1, there are three phases, one G1, S, G2. If you take a cell, in that cell there are three phases in interphases. In G1 phase, cell actively grows in size, metabolically highly active in this phase. If you take a S phase, here DNA replication occur. It is also called a synthesis phase. Histone proteins also synthesized in this phase. Here 2C becomes 4C. Amount is doubled, chromosome number still remains same, and centrosome duplication also occurs here. In case of G2, here cell prepare for its cell division. Protein synthesis, some kind of protein synthesis is occurs here prior to the cell division, which are essential for cell division. These are occurs in the cell division. We will go in a short way, not too long, because we have to practice MCQs as much as quickly. Now, as you can see, M phase is divided into mitosis and meiosis. As you know, in meiosis there is two types, meiosis 1 and meiosis 2. In case of mitosis is once occurs in only once. Here prophase, the phases of mitosis are prophase, metaphase, anaphase and telophase. Telophase. As you can see, this is cell. As you can observe, in this is chromatin. Now as you can see, this is cell, this is nucleus, in here chromatin. Okay, now what will happen? The nuclear envelope is disappear in the prophase, and the chromatin is also disappear, and it is condensed into a chromosome. Here chromosomes will form. In case of metaphase, as you can see, the chromosomes align at the equator. This is like this. This is called equator, and the chromosomes are aligned at the equator. The centrosomes on the pole side and attached to the chondrochores on the chromosomes. As you can see, in a phase, the chromosomes are moved towards poles by contraction of spindle fibers. As you can see here. Now if you take telophase, here the chromosomes are at the pole. Here chromosomes are you can see. Oh, sorry for the image, I draw it in a wrong way, but this is right one because it divides now. So chromatids are also divide. So that's why I erase it and I read right. Now in telophase, the chromosomes become uncoiled and become chromatin, and the nuclear envelope reforms. Cell organelles also reform in telophase. After telophase, here it comes cytokinesis. M phase is also called as karyokinesis because it is a nuclear division. Now, after the karyokinesis, cytokinesis follows. As you can see, in case of cytokinesis, sorry for the blurry video. As you can see, these are cells. One is assumed as animal cell, another as plant cell. The cytokinesis is two types in animal and plant cell. Here in animal cell, there is a furrow formation. And divides the cell. The furrow is outwards to inwards. Okay, as you can see, the furrow formation is outwards to inwards, and the cells are formed like this. If you take plant cell, there is a between the nucleus, there is a formation of fragmoplast. Middle lamella formation is happened here. This is also called as cell plate method. It only happens in plant cells. By this, these type of cells are formed. The cell plate method is inwards to outwards. In case of animals, it is outwards to inwards, as you can see. To remember the phases in mitosis, you can follow this trick. Pro met ana tilo. So remembering this, you can observe the sequence and you can easily guess. Now we are going to do meiosis. Here meiosis 1 and meiosis 2 happens. In between them there is a small duration called interkinesis is happened. As you can see, meiosis 1 is reductional and meiosis 2 is a similar to equational division. Here in meiosis 2, here in meiosis 1, duration of phases is high in meiosis 1, little long, not too high, just little long. As we get into meiosis 1, in meiosis 1, as you can see, there is a divisions which are similar to mitosis. They are prophase 1, metaphase 1, anaphase 1 and telophase 1 are happened, which are similar but quite different in this meiosis 1. First, prophase 1. Here the as you can see in prophase 1, it is divided into 5 types because the variation in uh, genetic material is happened there, so that's why it is divided into 5 types basing on the behavior of chromatin to chromosome formation and after that the chromosome behavior. As you can see, for remembering prophase 1 stages, this is the trick: Leiji, Padi, Dia. By this, you can remember any sequence. First, leptotin. Second, pakitin. No, zygotin. Sorry. Third, pakitin. Fourth, diplotin. Fifth, diakinesis. In pakitin, chromatin condensation is happen and chromosomal formation is also happen there. In leptotin, chromosomes are appeared. As you can see, in zygotin, chromosomes are come to each other which are homologous to each other. Homologous chromosomes are similar in size and appearance but differ in genetic material. When they come closer, there is a formation of centromerial complex is formed between them. In pakitin, where homologous chromosomes are attached by synaptic complex. In between them, there is a formation of recombination nodule, in which by formation of recombination nodule, the recombination process has done in this phase. For this, the enzyme recombinase is necessary. Here, at the cross, you can see homologous chromosomes crossing each other by exchanging genetic material between the arms. By this, by meiosis, genetic variability happens by exchange between arms of the chromosomal material between the homologous chromosomes. In diplotin, in zygotin, you can see synaptic complex is formed. In case of diplotin, the synaptic complex is dissolved or broken or breakdown. Here, homologous chromosomes are visible, but there is no synaptic complex here. But there is a formation of X shape which is called as chiasmata. In case of here, you can see the X shape is formed. It's still attached there. So it is called as chiasmata. Diakinesis. In case of diakinesis, there is this X-shaped structure is terminated, or you can say it like this: termination of chiasmata. By this, the homologous chromosomes are get detached by each other. In this stage, the nuclear envelope is disappeared, and the remaining cell organelles also disappeared in this phase. Until diakinesis, the nuclear envelope is surrounded. So, by means that from leptotin to diakinesis, the chromosome is happen. Chromosome is exist, but in case of diakinesis, the nuclear envelope will disappear. As you can see, metaphase 1. Here, chromosomes align at equator, but here homologous chromosomes are appeared as tetrad or bivalents. So this is called as double mesic plate. Here the spindle fibers are also attached and the centrosomes are at the pole side. This will happen in metaphase 1. After this, in anaphase 1, here if this assumed as cell, the spindle fibers attached to homologous. Each spindle fiber is attached to here. Each spindle fiber is attached to one chromosome. So the homologous chromosomes are divided or distance goes to the each pole. So each homologous chromosome goes to one of the pole. Here is after this telophase 1. Here you can see the chromosomes align at the pole, and the chromosomes are become uncoiled and become chromatin. After the nuclear envelope reforms. After that, we will study about the meiosis too. So I'm sorry about my English language and speaking skills, but this is a class. Here subject only matters. I think maybe. Okay, thank you. Now, as you in between meiosis 1 and meiosis 2, there is a short duration of interkinesis is exist, where the centrosomes are again duplication is occur in interkinesis, because as you can see when meiosis 1, as you can observe, uh, two centrosomes are goes to the each pole. So in meiosis 2 there is a need of another centrosome. So in the near interface, sorry, interkinesis, the centrosome is also again duplicated, so that it will useful in the meiosis 2. Now as you can see in meiosis 2, which is similar to mitosis. In meiosis 2, well, prophase 1, as you can discuss here, as you can draw the cell. Here as you can see, these are chromosomes are appeared. In metaphase 2, the chromosomes align at the equator and spindle fibers are attached to it. Here each spindle fiber is attached to kinetochores of the same chromosomes, which means for each chromosome there is two kinetochores and assembly of two spindle fibers is occur. In case of anaphase 2, here centromere splitting is happening. In case of anaphase 1, there is no splitting of centromere. In case of telophase, the chromosomes goes to the pole and becomes discrete elements or chromatin, and after that nuclear envelope reforms and cell organelles also appeared. After this cytokinesis happens. As you can see, difference between mitosis and meiosis. First, Here, chromosomes remains number is remains same. In case of meiosis, the chromosome number becomes half. Second, cell repair. Cells are repaired, replaced. Cell growth, no sorry, tissue growth. It is essential for tissue growth, cell replacement, cell repair, tissue repair. These functions are correlated with mitosis. Mostly mitosis occurs in all vegetative parts. In case of meiosis, it only occurs in sexual related organs. As you can see, mitosis, there is no genetic variability in mitosis. As you can see in meiosis, there is genetic variability happens. By genetic variability, it will leads into evolution. It is also important to maintain chromosome number in generation to generation. These are the differences. Now we get into the MCQs questions. You have to focus on MCQs practice and PYQs practice. This is a question bank. There is no answers here. As we get into the question, first one question, aiims usually occurs in eukaryotic cells, meristems, sperm cells are eukaryotic cells. Except for the B, option B will be right answer for this question because that is a prokaryotic cells. Option B here right. Now second question, which one is correct sequence of a cell cycle? Okay, we have to observe G1 as G2 and after that M follows. So as you can see, option C will be right. After the question number 3, you have to identify this picture. As you can see, as you can observe, this is G1, GS, G2. By this will be G0. As you can see, according these options, you can observe A G0, B G1, C S, D G2. As you can see here, when a cell comes under S phase, it has to go through the G2 and has to be done cell division, because S phase, after S phase, it is like unstable stage. In G0 phase, the cell exits from G1 phase, so there is no cell division until it is called on. So as you can see by these options, the answer is A. Now fourth question, each cell divides once in approximately every 90 minutes. It is also in NCERT, so option A. If you take another question, human cells, first to remove this, we'll erase this board. Okay, then now question number 4. As you can see in fifth question, the answer is 24 hours. So option B will be right. Sixth question, which phase occupies the maximum part of cell cycle? Here mitotic, meiotic or comes under M, so C is interphase is correct. If you see the phase cycle is a period of intense synthesis and growth, it constitutes 94% of the duration of cell cycle. It is interphase. Option A. The remaining options are comes under M phase. As you can know, option A is correct. Now, question number 8. Read the following statement about cell division and select the correct ones. M phase represents the phase when actual cell division occurs, and interphase represents the phase between two successive phases. Okay, in the 24 hours average duration of cell cycle cell division proper lasts for only about an hour. M phase constitutes more than 90% of the cell duration. No, option 3 is looks like wrong. First we have to write it like this, and then we mark for the right answer, just for some rough work. Here option 1 and 2 are correct, option 3 is wrong, so option A is correct. 1 and 2 are correct. If you look into the 9th question, which are the following phases of the cell cycle is not part of interphase? So option D, M, because remaining are comes under interphase. Question number 10, a cell cycle includes interphase and M phase, prophase, metaphase, anaphase, telophase, G1 S, G2 phase. Here the answer is A, because a cell cycle includes interphase and M phase. In which stage DNA replication takes place? It is S phase. So option C. Synthesis for hostone proteins occurs in, it is also happen in S phase. So here options S phase is not available, so we can take it interphase So which is also included, right? So we take option B as right answer. In interface, it happens in S phase. So option B. Let me erase this. Now question number 13. Select the incorrect, wrong one. Statement regarding S phase of interface. It occurs between G1 and G2. Okay. DNA replication in the nucleus in this phase. Yes. Centrioles duplicate in the cytoplasm. Yes. As DNA is doubled, the number of chromosomes also doubles. No. Remaining answers are correct. As you can see, as DNA double, number of chromosomes also doubles. No. Chromosome numbers remain same. So option D is correct. Now question number 14. The DNA content of individual cells and the number of cells in each phase of cell cycle can be determined using flow cytometry. Which of the following combination of phase of a cell cycle and its corresponding DNA content can be considered normal? Diploid cells found in the G0 or G1 phase, cells with twice the normal DNA content in the early M phase. Okay, cells with intermediate amounts of DNA in the S phase, cells with twice the normal DNA content in the G2 phase. As you can see, this question is one of the tough, I think. Found in the G0 or G1. First one, right? Cells with twice the normal DNA content in the M phase. Okay, cells with intermediate amounts. So here option A is correct because it is also same. So all the options are correct here. As you can see, if you take a graph among amount and the DNA content, DNA, which is both are same, and amount was just phases. You can see G1. After G1, S phase is fluctuating, which where 2C becomes 4C. After the G2, after this M phase happens, where 2C becomes 4C becomes 2C. DNA content is become half. As you can see, 15th question. Cell would normally proceed to mitosis without interruption. Oh, sorry. Cell would normally proceed to mitosis without interruption. Once it has entered the S phase, yes, this is the right one. Once it has entered G2 phase, if S phase enters, that it will basically become unstable and it has to go to the G2 phase, as you can see. And after that, it goes into the cell division. Sorry, I am dubbing this video. I know my audio and that video is not syncing sometimes, but it's okay. We want the content, not these silly things. Okay, now the answer is A. Once it has entered the S phase. 16. The cells that do not divide for the exit G1 phase to enter inactive stage is called G0 phase. So here option D is correct. Now, okay, now we get into the 17th question. Which of the following statements is correct? Animals can show mitotic division with haploid and diploid cells. After S phase, the number of chromosomes becomes double. No. During G2 phase, proteins are synthesized in integration for mitosis while cell growth continues. As synthesis phase marks a period during which RNA synthesis phase, then cell growth.